Hi everyone, my name is Savannah Perry. I'm a fourth year marine science student.
And my name's Cameron Williams. I'm a fourth year UF biology student.
We chose the paper Microfragmenting for the Successful Restoration of Slow-Growing Massive Corals.
So why did we choose this paper? I initially became curious when I saw in our chapter in Sex and the Sea that researchers in Florida had developed new techniques for propagating corals as part of their conservation efforts. So naturally, I was curious to find the actual research paper behind this information. And when I did, I was really interested to find out about the overall research project and their goals here. Um, and one of the important goals of this project is to find ways to propagate large stony corals. Since these grow really slowly, but they're really important ecologically as reef builders, as the like 3D habitats that we've learned about that provide shelter for other marine organisms. Previously, the corals that had been selected for restoration projects were just a few species, and they were really easy to grow. So understandably, they were being selected for these propagation experiments, but they're not necessarily resilient, especially to the high temperature events that cause coral bleaching. And so these stony corals are resilient to thermal stress, and so they're important for the long-term restoration projects so that the corals that they propagate and outplant will be resilient in the future as sea temperatures rise due to climate change. Another reason is simply that the research takes place in Florida, and so it's close to our homes and our hearts, and involves ecosystems that we're familiar with in some cases. And so I just did a quick Google map to show how you could get down to Summerlin Key from Gainesville, um, um, and the research station where this research took place is down there in Summerlin Key. There's also a base campus and an aquarium in Sarasota. So the experiment focuses on two corals, the mountainous star coral, Orbicella faveolata. And then the second coral is Montastrea cavernosa, or the great star coral. And the interesting thing about this coral is that the polyps are actually like the size of a human thumb, and the tentacles come out, fully emerge at night. So you can really see the like polyp shape of the coral, and you can also get a sense for truly how big these corals are. Another interesting aspect of this paper, I thought, was that the propagation methods were sort of counterintuitive. They discovered that these smaller fragments would grow faster than larger fragments. So this, invites us to speculate on why that would be the case. Like, what about the growth process or the reproduction process of corals would lead to this result? Another reason why this paper was interesting is that it was published a few years ago, but there's already been some important follow-up research based on it. And most importantly, just a few months ago in August, the Motes lab announced that these corals had already reached sexual maturity. So not only were they growing really fast from these microfragments, but that some of them were now producing gametes. And so the mountainous star coral in particular was the first species of these like large stony corals that had been documented to produce gametes after being restored in Florida and the Caribbean. And they did this with an additional species too, the staghorn corals. reached maturity in record time. And so you can find a couple great videos on the Mote site where the researchers from this paper announce these new results.
So there were 3 authors for this paper. The first author is Christopher Page. He's a biologist at the Mote Marine Laboratory in Summerlin Key, Florida, which, as Cameron mentioned, is the satellite station for the Mote Marine Laboratory in Sarasota, Florida. There, they typically focus on coral research and restoration. He has 8 publications which deal with coral restoration and ecology. The second author is Erin M. Muller. She has a bachelor's and a master's in marine biology from Florida Institute of Technology, and she also has a PhD in biology from Florida Institute of Technology. She's the program manager and science director of the Mote Marine Laboratory in Summerlin Key, Florida, and her research typically focuses on coral health and disease dynamics, especially in relation to climate change and ocean acidification. The final author is David E. Vaughn. He has a PhD in botany and plant physiology from Rutgers University. He's an aquaculture scientist, and he's the director of Mote Marine Laboratory in Summerlin Key, Florida. He's also the founder of the 60-acre aquaculture development park at the Harbor Branch Oceanographic Institute in Fort Pierce, Florida, which focuses on training, demonstration, and education in aquaculture science techniques. So he's a very important aquaculture scientist, which is an ever increasingly important field.
Okay, so getting into the paper itself, the overall purpose was to improve these coral gardening techniques, which is another word for these restoration, propagation restoration techniques. So the researchers wanted to include some more species, these large stony corals, and try to achieve better growth rates, which is especially important since these large stony corals grow so slowly. In the past, really large fragments were being used, which also limits the scale of these propagation projects. So if you're wild collecting corals with the intent to propagate them and outplant them in new locations, you want to be able to like produce as much new coral tissue as possible. So that's one of the overall goals. Another goal was to have a longer term experiment. Typically, coral gardening was not monitored for more than 1 year. You can speculate on the types of sort of material, economic constraints cause that to be the case. But for this experiment, they were able to monitor it for over 2 years, well over 2 years. So their research question was specifically, will microfragmenting corals, which basically means like cutting them with a saw into tiny pieces, into 1 square centimeter pieces, increase the survival and growth compared to the larger pieces, the traditional sized 16 to 24 square centimeter pieces. So their hypothesis was that the microfragment arrays would outperform the single larger fragments. Arrays refers to the way that they glued them onto a tile. That's called an array. So they wild collected these corals and then fragmented them and raised them in a lab where they could, control the oxygen, nutrients, that sort of thing. And they detail all of those parameters in the paper. And they raised them for 1 to 2 years for the large pieces and 6 to 12 months for the smaller pieces. And so they ended up with 12 large fragments and 96 microfragments. And then they planted these in 2 different environments, a nearshore environment was shallower at 3 meters and an offshore environment that was deeper at 6 meters. And in both places, there were dead corals where there had been previous coral bleaching events. And so they glued the new corals to the dead reef using marine epoxy. And then in order to gather data on them, they used top-down photographs with a little tile next to them for reference in order to measure changes in surface area. And they also monitored them for like grazing and damage by fish and other organisms. And there were 2 bleaching events during this time period. So they used the photographs to monitor the corals for color, just using a scale from 1 to 5. So the initial results were really exciting. In the nearshore plantings, the microfragments produced much greater growth, up to 6.5 times more growth, but in one species, but overall in both, there was a lot more. In the offshore plantings, there's a lot of heavy predation events that interfered with the experiment. And so the long-term comparison wasn't actually possible because of the limitations in the experiment, including these predation events, and just including the kind of small sample sizes. There wasn't, they saw good results, but they weren't like statistically significant. And so nevertheless, these initial results were really exciting. So for example, if you remove the predated corals from the data, it shows that the growth rate was 10 times as high for the microfragments as for the traditional fragments.
And finally, we have some discussion questions for you. So the first one is, do you have any ideas on how predation of microfragments could be reduced? The second one is, were there any limitations in the methods? The third one is, what factors influence growth rates in microfragments? And the fourth one is, what are some future studies to expand on this research?